Okay, why don't we get started today? We're going to try to finish up enzymes today, and then we'll move on to the core material of the course, which starts in the nucleus. But for the moment, we're using enzymes as sort of our poster child of protein function. And that chapter on proteins covers lots of different proteins, so we could spend a lot more time on this one chapter, but we're actually going to pick up all those protein functions as we go along. So this is the one that we're getting used to various properties and how you measure them, and it actually ties into the departmental seminar that was given yesterday by Dr. Kobos, who's an oncologist over at the Health Sciences Center. We're not just doing these enzymatic assays and studies for fun. It actually, a lot of tumors have enzymes that are overexpressed or mutated that cause cancer. And so one of the more effective ways to knock down cancer cells and kill them is to be able to inhibit the enzymes that produce the cancer. And so pretty soon we're getting into the era of, if you come in late, please come in through the back door. Thanks. To an era where you'll go in if you have if you're suspected to have cancer, they will sequence your entire genome and identify all the mutations and all the proteins that are, or genes that are underexpressed or overexpressed, and find the ones that are mutated and come up with a cocktail of inhibitors that will take care of your cancer. Because your cancer is going to be different from anybody else's, even if it's breast cancer or prostate cancer. Within that group, there's a lot of heterogeneity. And so one of the things that drug companies are looking for are small molecule inhibitors of enzymes that are overexpressed in cancer. Now, we talked about one poster child that's like that. That's C-SART. That's a tyrosine kinase. And so this is not trivial stuff, and so there's some basic elements that we need to get down because it's important. And we'll use those ways of measuring things not only for this subject, enzyme control, but also for many other types of proteins like membrane transporters. Okay, so we only have to learn it once. And so where were we? We were talking about basically how do we quantify enzyme kinetics, and we'll finish that off, and then we'll talk about how our enzymes regulate it. Okay, so the essence of what you needed to know from last time were these two components of the Michaelis-Menten equation. Okay, because that's what we're after when we try to derive the characteristics of some sort of enzyme. What do those things tell us about that enzyme? Number one, Vmax tells us two important pieces of information. How many enzymes are in that cell? Okay, so that in terms of cancer, what you're asking is that protein overexpressed or underexpressed? Okay, so that's one property. How many are in that particular cell? And the second question is the K-cat. Remember, we get that K-cat is equivalent to the K2 rate constant. It is the rate at which the enzyme substrate complex is converted to product. Okay, so it's the slowest step generally. So it's a small number compared to K1 or K minus 1, and it tells us the fundamental rate at which that enzyme can take a substrate and convert it into product. I like to use, I won't go into the depth that I normally do, an analogy of monkeys shelling peanuts. Okay, so we can actually learn a lot from just thinking about it in very simple terms. If a monkey is hungry, it's going to shell a peanut and eat it, right? Okay, so consider that the monkey shelling the peanut is the enzyme. And the peanut is, of course, what? The substrate, right? So there's a couple things to determine how many peanuts the monkey can shell per second, right? It's the turnover rate, and that is if he's sitting there with all the peanuts in the world sitting around him, and so he doesn't have to get up and go collect peanuts. So that's no longer a limitation, and that's what happens at high concentrations. The enzyme is completely saturated with peanuts, and so is the monkey. Okay, so all he has to do is pick it up, and picking it up represents what? K1. If he drops it, that's K minus 1. Okay, K-cat is how long it takes him to shell it. And so the thing, there's an intrinsic rate at which all monkeys shell peanuts, and that's called the K-cat, and it's true for that enzyme and that protein and that isoform. Okay, if you mutate the enzyme, you can change K-cat. But just increasing the number of enzymes does not affect K-cat because it's the fundamental rate at which one monkey shells peanuts. Okay. And it's also, so what we're really talking about is, so how else can you affect that reaction rate? Well, you can decrease the number of peanuts. Let's say we put one peanut in that corner, and one peanut over there, and one peanut over there. The rate at which that one monkey shells peanuts is going to do what? It's going to plunge, because he's got to walk over there, and he's got to walk back there, and he's got to walk over there. That represents the collision rate of substrates with the enzyme. The lower the concentration of peanuts or substrates, the lower the collision rate hitting the enzyme. And so that's the effect of concentration on enzymatic activity. It always speeds it up if you increase the substrate concentration. Okay? But if it's all piled up on top of him, he doesn't have to walk anywhere, and so he's going to shell at a rate that's equal to his kk. Okay? All right, so we now know, and so what's the other way, besides affecting concentration, how can we speed up the number of peanuts shelled in the room? What else can we do?
Increase the number of peanuts?
No, peanuts increases the concentration, so we've already dealt with that. That is, absolutely, never forget that. That's called, a substrate, it's a substrate effect. It's called mass action, actually, when you increase the substrate to make the reaction go faster. What else could we do?
Make the peanuts easier to peel.
Pardon me?
Make the peanuts easier to peel.
Okay, but that's the K-cat, and so that's an intrinsic function of that monkey. He can't go any faster than his intrinsic rate. We could mutate it, right? And so that you can do that if you change the protein's function. But we're talking about just leave the monkeys the same. The number of substrates are the same. What else could we do?
Increase the number of monkeys?
Yes, we can let more monkeys into the room. And so that's why this is the product of the number of monkeys in the room and the rate at which each one shells the peanut at its fastest rate, with being saturated with substrate. Okay, now, so what does the Km represent? The Km really represents how fast the monkey can pick it up without dropping it, and it's also affected by the rate of catalysis. But that K-cat is usually so small that we just focus on this, okay? And so if the monkey picks up peanuts really efficiently, but only every once in a while drops one, that's going to be that makes this a very small number, doesn't it? Because the denominator gets big, and the numerator, the rate at which he drops them, is very small, okay? But what if he's sort of a retarded monkey, And so for every 2 that he picks up, he drops 1. Okay, so that's going to increase the numerator, k minus 1, and the k1 decreases. So that becomes a big number. That means his affinity is very poor for the peanuts. Okay. So all of these tell us something very intrinsic about the protein's property and the way it catalyzes stuff, so it's important stuff to know. Okay, do we have any questions about that? Okay, we'll test ourselves as we go along to see if we understand this very basic idea. The next thing we need to do is go briefly over how would you actually do this in the laboratory. You're actually going to do it in Cell Lab, but we'll go over here briefly. Okay, so we want to know, how do we quantify enzyme kinetics? We have to be able to do that in the laboratory, obviously. And so what we're after these 2 parameters, because we know the substrate concentration, and you need to know this equation, Collie, obviously. We know the substrate concentration, but we need to get these by a curve fitting program, okay? And so let's do that. And so here's an experiment where we have a series of test tubes, and and I'm going to try to use this over here, and I want people in the back to let me know if it's too small, because I'll have to move it back over here. And this is getting in everybody's way, and I have to change screens, so I'm going to try it this way. Okay, so the reaction that we're going to look at is glucose, okay, plus ATP. That's the phosphate donor, okay? And it's going to phosphorylate that glucose molecule at the 6th position. And that enzyme is known as hexokinase. Is that reasonably in? Hexokinase. And this is a family of enzymes that there's lots of so-called isoforms. It means they all do the same thing, but they have slightly different characteristics in their affinity for glucose and ATP. Okay. And I'll give you a good example of that later as we go along. But what it does is it turns this into glucose 6-phosphate, and then obviously you rob the ATP of a phosphate, so it's you make ADP. So that's the reaction. Now, if we're measuring it over here in this series of test tubes, we have to decide this. There's 2 substrates, correct? There's the phosphate donor, which is ATP, and then there's glucose. So you need to focus, to make this simple, like we do in our laboratory, we focus on one substrate at a time. So what we'll do, let's focus on glucose. How can we get rid of ATP's effect in this series of test tubes that we're going to be measuring the rate? Somebody said, what is that?
Saturate the mixture with ATP.
Absolutely. So it's saturate, but a very high concentration of ATP, so it's no longer determining the rate of the reaction. Okay, so we're going to keep ATP concentration the same in every single test tube, but we're going to vary the concentration of glucose because we have complete control over that, right? So we're going to start with 0 and then increase it as we go along. Okay. Now, what do we do with we have to put the enzyme in there, so we're in control of the amount of enzyme. But what we do is we put the same amount of enzyme in every tube. We do not change the concentration. You have to do it at the same concentration because you won't get a Michaelis-Menten curve if you vary the enzyme concentration. Because now you have 2 variables, right? You've got the substrate concentration, and the enzyme concentration is going to change the what parameter? Km or Vmax? The Vmax is going to change the number of monkeys in the tube, right? Enzymes in the tube. Okay. All right. So we're going to leave that number constant, and then we're going to measure the rate of glucose disappearance. Now, glucose disappearance is going to equal the appearance of glucose 6-phosphate. So you can measure either one, either the rate of appearance or formation of glucose 6-phosphate, or you can measure the disappearance rate of glucose. And so we'll do disappearance rate. And when do we measure it? We don't do it just willy-nilly. This is, remember, an initial velocity. So we're going to look at our equipment. Let's use a spectrophotometer, and we're going to a very sensitive instrument that reports back to us the change in glucose. And we're going to measure it at a very early time point. In fact, the earliest time point at which we can statistically detect at a very good error rate the concentration of glucose. Okay? And so we're going to choose that very early rate. That enables us to use the Michaelis-Menten equation. Otherwise, we have a very complicated equation that we would have to solve. Okay? So you have to do initial rate as a function of glucose concentration. So this is the plot. Always look at the axes when you're shown a graph. That's the first thing you should do is what's being measured here, what's the dependent variable, and what's this variable. Okay? And if you know that, what you find is if you do the math, you get these values here, and you can do mathematical ways of deriving Km and Vmax, logarithmic transformations. It's no longer necessary because we have things called computers, and we fit the data to the Michaelis-Menten equation. Okay, so we're going to fit this data, these time points, the glucose concentration, to this equation, and it will spit out Vmax and Km. Okay. All right, so is there any question about how we're actually measuring this? Okay, good. And you guys can see that okay? Okay. Okay, so ask yourself the question. What happens if we now repeat the experiment and we put twice as much hexokinase in each tube? What's it going to do to those parameters? You should a priori be able to determine what the effect is. Yes.
Would Vmax be double?
Vmax would be double. It's a linear function of the number of monkeys in the room, or the number of enzymes in the test tube. Excellent. That's all it does. Because we're not changing the rate at which the monkey shells peanuts, we're changing the number of monkeys in the room shelling peanuts. So we're not affecting K-cat, correct? We're affecting Vmax only by shifting the total amount of enzyme. Okay, so it doubled. What about Km?
Dan would not be affected.
Km was not affected, correct? Because think about the equation. It's K minus 1 plus K cat over K minus 2. You're not affecting K cat, we already said that, because you're using the same monkey in each tube. And you're not changing the intrinsic K1 and K value of infinity for the substrate, because you're not changing the enzyme isoform. Okay? So that's where I want you to be able to look at this and predict what should happen. Okay. Okay, so I guess that brings us to the topic of enzyme regulation. Now that we know how to quantify stuff, all right, so good. We have a couple of different kinds of inhibitors that we can talk about. There's two types of inhibitors. Basically irreversible, that is, when these compounds bind to the enzyme, they basically poison it, and they usually form some sort of bond with the protein, and they don't let go. Okay, and the only time we really use those is when we're trying to kill something like each other in gas warfare, or we're trying to kill insects. Okay, so that's not a good way. That's not a good thing or drug to devise for treating a disease. It's too deadly. Okay, so the other version is reversible. Okay, and it's usually forming transient bonds with non-covalent bonds with the enzyme and affecting its function. Okay, and there's two basic categories that we're going to talk about. This is a really actually very complicated area of biology. It's called, or biochemistry, called enzymology. We're going to look at the very simplest cases, because if you can understand that, the rest is something you can pick up. Okay, particularly if you take an enzymology course. Okay, so we want to know what's competitive versus non-competitive, and they really do look to make a lot of drugs competitive inhibitors of the substrate that they're trying to knock out. Okay, and so what's competitive inhibition versus non-competitive? Okay, so the hallmark of competitive inhibition, actually there's a good diagram in your book before we get to that. Right here. Okay. So you have the enzyme, and what it's usually going to do is pick up the substrate and convert it to a product. Okay. Good. All right. Now the competitive inhibitor binds to the active site, and not only that, it binds to the substrate binding site within the active site. It's trying to mimic the substrate. But the problem is when the enzyme binds to that inhibitor, it can't break down that substrate. It's a fake substrate. Okay. So how is that going to affect the enzyme kinetics? So that's what we're going to do over there here in just a couple of minutes. The other way of inhibiting things is to take an inhibitor, and instead of binding to the active site, it binds to another site away from the active site. And basically it poisons that enzyme. It binds to the enzyme and affects its shape. What's that going to do to the function? It's going to affect the function if we affect the shape. Okay? And typically what it does is it distorts the binding site, and actually not the binding site so much as the ability of that enzyme to catalyze. So it really makes K-cat go down. It's really small to the point it's almost zero. Okay, so the ones it binds to, it poisons. Everything else that it doesn't bind to is left intact. Okay, so you got two different things. How much of that fraction of enzyme has been poisoned is really what you're asking. All right. So let's go over here and do the kinetics. And these kinetics only apply to enzymes that are Michaelis-Menten enzymes. Okay. If it's not Michaelis-Menten enzyme, it's generally what's called an allosteric enzyme, and these equations don't apply. So it depends on the type of enzyme. So that's the first thing. Are you dealing with an enzyme that follows Michaelis-Menten kinetics? Well, how would you determine that? First, you got to do it without the inhibitor, right? And you should get a square hyperbola, right? Okay, so let's assume that's what we get. Let's do this. And we're going to do, obviously, initial velocity as a function of substrate. this. And if this is Michaelis-Menten enzyme, it should do this. Okay? And from that, we can fairly easily, so this is the enzyme in the presence of no inhibitor. So there's no inhibitor in the test tube. Okay? And so we should be able to come up with an asymptotic value of Vmax. And we should be able to go down here halfway and go over here and come down here and get the Km. Okay? All right, so we do that. Now, let's put the inhibitor in the test tube. And we're going to put a small amount of inhibitor. We're not going to put a lot of inhibitor. Okay? We're not going to saturate it. Small amount of inhibitor, but we put the same amount of inhibitor in every tube. We're not varying the inhibitor concentration. Okay? If we do this, what happens is we get this curve. And what happens is the Vmax is the same. The Vmax with the inhibitor present equals the Vmax without the inhibitor. Okay, well, let's think about why that would be the case. So you come down here, you come over. Oh, but look what it does to the Km. It makes the Km in the presence of inhibitor increase. So what is this telling you about the effect on the enzyme? If we understand what Km means, kind of, right? So if we increase Km, what does that say about the enzyme? Does it change the number of enzymes? It's not working as well. Okay, what's it? But technically, remember what we said about what Km really reduces to. Let's say that Kcat is really small. I'm sorry?
K minus 1.
Right, and so what that number is the dissociation constant. So it's inversely related to the affinity. So if we increase Km, if Km gets big, that means the affinity gets small. That means it's crummy. So the effect of a competitive inhibitor is to cause the monkey to drop the peanuts. Instead of picking up 10 and dropping 1, it picks up 2 and drops 1. Okay, so it has a really crummy affinity, right? So that's the effect on the enzymatic kinetics that a competitive inhibitor has. And that makes sense. So, but why does it not affect the Vmax? Remember, they're competing for binding up there to the active site. And what's going to happen at high concentrations of substrate? Let's go to infinity, all the way out there. Who's going to win the battle? Let's say we got a monkey shelling peanuts, and we got a mountain of normal peanuts, and we got one ringer in there. Most of the time he's going to be picking up what? Regular peanuts. And so that's why it doesn't affect the Vmax, because at high concentrations of substrate, we outcompete the inhibitor. Right. Good. So that should be intuitive. So this is competitive. And the first thing we had to do, though, is we had to measure that normal kinetics to see what it looks like. And if it looks like a square hyperbola, we can probably use the Michaelis-Menten equation. Now, let's redo the experiment, and this time we're going to use that type, the poisoners, okay, the non-competitive. Non-competitive inhibition, so yes, that's what we do. We measure it again, and we get initial rate versus substrate concentration, and we should get this, okay? And so we should be able to come over here and get a Vmax. Come down here halfway, and we get the Km, because the Km definition practically is the concentration at which the rate is half maximum. Okay, we know that. Now, we're going to put that inhibitor in there, and lo and behold, this is what it looks like. All right, so you can tell right away if we plot this, Vmax has decreased. So the main effect of a non-competitive inhibitor is to kill some of the monkeys in the liver. That's why it's decreased. Some of those are poisoned by the enzyme. Okay, but it leaves the rest of the monkeys, although there's only half of them, still shelling peanuts at their traditional rate, correct? And so if we come over here, if I did this properly, what happens? I come down halfway, come over here, it should end up being the same Km in the presence or absence of inhibitor. Okay? So the hallmark of non-competitive is no effect on KM, because all the monkeys that are not inhibited are still doing their normal thing, but half the monkeys in the room have been killed off. And so V max is decreased. Okay? Questions about that? Okay. We'll do some practical examples here in a few minutes to make sure we're all on the same page. Because it's easy to go, yeah, I'm pretty sure I understand that, but when it gets mixed in with the other stuff, it can be a little confusing. So. Okay, so there's a couple of different ways that our cells have put together enzymes to speed things up, okay? The problem, so one thing we could do, as someone mentioned, is that we could increase the substrate concentration. That would speed up a reaction, correct? But we have so many reaction pathways, biochemically, it doesn't make sense to have large concentrations of substrate for every single reaction. So that's a poor way. Nature does not do that. In fact, usually the average concentration is very close to the Km of the enzyme. Now that's a generalization, but usually those concentrations are micromolar or below. They're not high. So we're not speeding up reactions because we increase the substrate concentration. Would that actually work? Absolutely. But we don't do that in a cell. Instead, what we do is we can effectively increase the concentration by two mechanisms. We can take a complicated, this is one big enzyme. It's a multi-protein complex that I mentioned last time. It's this pyruvate dehydrogenase. What it does is it works in the, what do you have to know about a protein? You have to know where its location is. It's in the mitochondrial matrix. And when pyruvate that's come from glycolysis comes into the mitochondrial matrix, it splits off one carbon and you blow it off as CO2, right? And then it attaches it to coenzyme A, and so you make acetyl coenzyme A. So that's that reaction. But that requires a bunch of, 3 different types of enzymes. But since they make a sequential product, in other words, the product of this enzyme is handed over to this enzyme, and the product of this enzyme is handed over to these guys. So they're all in one little place. So guess what? The product of this reaction doesn't have to diffuse anywhere. It's right there already. So we call this reaction, this is a way to defeat diffusion-limited reactions. And what that means is all these enzymes, just by themselves, are so good at what they do. The thing that limits the rate is the time of diffusion that the substrate takes to bump into the protein. That's a really good enzyme, okay? And so that's the case here. So if it's diffusion limited, one way we can speed up the reaction is to put all of the sequence of enzymes together in one complex. And that's how you solve that in your cell. So you'll see that a lot of complexes for splicing RNA, for any metabolic reaction, they're assembled into a complex. Okay. Now, the second way that we can sort of defeat the concentration problem is we can put it into a compartment, a membrane-bound compartment, and thereby increase locally the concentration of substrate. So compartmentalization is a way of speeding up reactions. Assembling protein enzyme complexes is another. Okay, so that's a good way to do things. Okay. Now, your cells have their own little inhibitors that they're always producing. Now, there's not very many of them that are actually competitive and non-competitive. They tend to be what are called allosteric effectors, or the term allosteric regulators is also true. Allosteric means that the regulator binds to another site. Okay, it's somewhere else generally, and not in the substrate binding site, although there's problems with that definition. So it comes from allo, which other, okay? And so what's going to happen is we're doing this reaction here, okay? And then we make B, and then we go, the side reaction is to make Z, okay? And what we don't want to do is, if a lot of Z builds up, we want to slow that reaction down because we don't want to overproduce the amount of Z. So this is a self-controlled reaction. So this is called feedback inhibition because Z comes back and inhibits the enzyme that converts B to X. Okay? Feedback inhibition. And most of these enzymes that respond this way are allosteric enzymes because they have a binding site on their surface for Z. Okay? And then you can actually come up with a really elegant series of reactions and do this many, many different ways. And so you have a series of enzymes that not just have one allosteric regulator, they have multiple. You'll see multiple binding sites for feedback to those enzymes. So if it's a critical enzyme like it is for aspartate, can lead to lysine, homoserine, isoleucine, and methionine. And we don't want to make too much of this stuff here. Okay, so if we build up any of these three components, they feed back and slow this reaction rate. Okay, because we need aspartate for other reasons. Okay, so that's the whole idea. So we're going to look at the anatomy of this type of allosteric regulation, and it's very different from Michaelis Menten. Yeah.
So the dreaming, it can inhibit
There's-
homoserine.
yeah. Oh, there we go.
It does it in both in two different ways.
Yeah.
How is that controlled? Or
Well, it's double. It's redundant control.
okay.
Yeah, because it means that's really dangerous to make too much of that stuff.
Okay.
Usually that's what it implies. So you not only have feedback of three different inhibitors, but each inhibitor can simultaneously be controlling several reactions too. So it gets very complicated. But let's take the simple case, and you won't think it's simple, but we will. And we're doing this now. How are enzymes regulated? And we're studying allosteric regulation. So one of the most studied allosteric regulators is this enzyme called ATCase. And I'm not going to make you say it by its real name, which is aspartate transcarbamylase. Okay, we'll just call it ATCase, right? And there's absolutely no reason to memorize this reaction. This is not biochemistry class, okay? So I know you'd love to do that, but take a moment to commit it to memory, and let's just move on. Okay, so this is the reaction. We're converting N-carbonyl phosphate to N-carbonyl aspartate, and it's going to be used to make nucleotides. This is an important reaction, right? Okay, so let's look at how it's regulated. There is two types of allosteric effectors here. There's one CTP, cytosine triphosphate, will feed back and is a negative inhibitor of ATCase. So we know what that is. It's a feedback inhibitor, and it's an allosteric inhibitor. Okay, and then if you have a lot of ATP and you want to use it up, let's say we have an abundance of ATP, a substrate that is in a reaction prior to that can actually cause feed-forward acceleration. So we call that feed-forward activation. So ATP is an allosteric activator. It speeds up the reaction. Okay, so we have two effects. We have inhibition and we have stimulation. Okay, how are those going to independently affect this enzyme? So let's look at, we have to know a little bit about the structure of this enzyme. Okay, we talked about this, but ATCase is really interesting because it has 6 catalytic subunits. Each one of them are identical. In other words, they bind the same substrate. And then we have 6 regulatory subunits. So the thing that's detecting the activators and inhibitors is actually a separate protein that's attached to the multi-protein complex. And let's see, do I have a picture of it? Yeah, well, I'll get to that picture. Well, let's go do that so we know what we're talking about. This thing looks like a spider, kind of, okay? So we have the catalytic subunits. We have 3 on the top and 3 on the bottom, okay? And the regulatory subunits are sort of worn like a belt all the way around this spider, okay? And this spider can either be crouched down or it can be standing up on its legs, okay? The crouched down state is what we call the tense state. And so that kind of makes sense, doesn't it? It's tense, okay? It's also, that's another word for low affinity. So here, you need these terms, okay? So we've been using KM and affinity and monkeys, so let's don't get confused. So here's the rules, the naming rules, okay? In the tense state, it's also called the open state. It's also called the low affinity state. And your book uses interchangeably open intense. Okay, but they're calling it T. That's what the T stands for. Okay, now the R state is just the opposite. It's the relaxed state. R stands for relaxed, or the enzyme is in the closed state. Okay, or the high affinity state. All those words mean the same thing. And unfortunately, your book kind of switches back and forth, and you need to get used to it because different papers use different terms, and so you need to know all three of those terms in each set. Activation, or where it's highly active. Now let's go back and use that. And this example kind of looks like alligators to me. Okay, so what happens is we're talking about, what are we doing? It's going to stabilize the enzyme in the relaxed state, the closed state, the high affinity state. So it's actually going to, this is going to be an allosteric activator. And what it does is it binds to this alligator when it has its mouth closed and relaxed. Okay? And that's the high affinity state. And so you're going to speed up the reaction because you've stabilized the alligator with its mouth closed. Okay? So you convert all these open. See here, you've only got one bound, and you got 10 of them or so, or 9 of them are open. They're in the tense state. You know, the mouth is tense. It's open. It's ready to shut. Okay. Then you add the activator, and it causes them to shut, and it stabilizes it in the shut state. And so that's how we speed up that reaction. So this is an allosteric activator. And then we can do the opposite, of course. We can actually put something in there that actually causes it to go to the open, tense, inactive, or low affinity state. And so these guys are all barely, alligators are crunching on their food. You put the allosteric inhibitor in there, and it stabilizes them with their mouth open. Okay? Substrate falls out. Doesn't do its job. So it's inhibiting the alligator. A lot of it, this is a zoo of critters today. Okay? Monkeys and alligators, spiders, lions and tigers and bears. Okay. All right, so we know what happens here. So we take the example of CTP, which you know is an allosteric inhibitor. So which way is it going to convert it? So when you put the CTP on, you're converting it from the relaxed, closed, high affinity state to this tense, open, low affinity state. That's the effect. Okay? And where is the CTP binding? It's binding to the regulatory subunits, not to the catalytic subunits. We call that heterotropic allosteric regulation. And there must be a homotropic, too. Heterotropic. Because it means that the thing that's doing the regulation is not the substrate. It has no binding affinity whatsoever for those catalytic subunits. And that's a typical thing for an allosteric enzyme. So now the next question is, how do the kinetics look compared to Michaelis? This is not a Michaelis-Menten enzyme. So if you plot this, you do the reaction, you're going to go, I'm going to get Km and Vmax, okay? So you plot this, you do your regular thing, velocity, substrate concentration, this is what you get. That's not a square hyperbola. What is that? Sigmoidal. Okay. This thing conforms is fitted to the logistic equation. And we're not going to worry about the mathematics, but if you wanted to look that up, you could do it. But you cannot use the Michaelis-Menten. This thing has no Vmax in Km because it's not a Michaelis-Menten enzyme. It's actually called Bmax, and it's a 4-parameter equation. It's entirely different. So we're not going to just understand that you cannot determine a Vmax or a Km for an allosteric enzyme, except in one condition, that is if you break it apart. Now, what happens is, so this has got 6 catalytic, 6 cats, 6 regulatory subunits, okay? What's causing this thing to have a sway back? It's not the allosteric regulators. I haven't put any of those in the test tube yet. What's causing this swoop up, this S shape, is the multiple catalytic subunits. It's called positive cooperativity. The subunits are cooperating with each other such that When one substrate binds to one subunit, it enhances the affinity of all adjacent subunits for another substrate. And I'll show you how that works. Positive cooperativity. Okay. And guess what? You can have negative, but we're not going to talk about too much about negative cooperativity. Basically the way we explain positive negative cooperativity is you've got some substrate, and then you have an enzyme. Let's just use one that has 4 subunits. Okay. And so this enzyme, the substrate's going to bind to let's see, there's 1, 2, 3, 4 different sites. Okay. And what happens is site number 1 is bound, and what happens is you have 2, 3, and 4 that are unbound. But this changes its shape and affects its neighbors. It affects that one. It affects this one. It has an allosteric action on its neighbors. Okay? This is called positive cooperativity. And what it does is it converts all of them into the high affinity state. Let's say it changes into a rectangle when it's in the high affinity state. So what's going to happen is all of them are going to become simultaneously rectangles. This is the concerted model of allosteric regulation. And we're not going to go into you can have all kinds of different models, sequential models, but this is the one that's typically encountered. All right. So all it does is you bind to the first one, and that enhances the activity of the other subunits. And so what happens is it means over a very narrow range and concentration, you get a large increase in enzyme activity. That's the practical advantage of having positive cooperativity. This actually happens when your 4 different subunits of hemoglobin binds oxygen. When it binds one oxygen, remember hemoglobin is a heterotetramer. When it binds one, it suddenly gets high affinity for 3 other oxygen molecules, and so that causes the oxygen binding equation to look exactly like this. Okay, positive cooperativity. So it's multiple subunits, and when a substrate is causing this change in activity of the enzyme, this is called homotropic regulation. Okay. So the sigmoidicity is controlled by the positive cooperativity, but that's a form of allosteric regulation because we're binding to this guy, but it's affecting the shape of its 3 adjacent partners. So it's allosteric, isn't it? So that's when the substrate changes the structure of adjacent subunits, it's called homotropic. When it's some other different effector, like it's CTP or ATP that's not a substrate, it's called heterotypic. And it has a completely different mechanism. Because how does that change this equation? Okay, well let's talk about that. So how would a heterotropic allosteric regulator affect this curve? And then we're almost done with this. Okay, so here's the unfettered Vmax substrate. Okay, this is without any kind of activator or inhibitor. If we put an activator, and let's put that in green to remind us that we're activating. Okay, what it's going to do is it's going to shift the curve to the left. It's going to make an extremely high increase in enzyme activity over a very narrow substrate concentration. That's highly activated. Okay, so that's if we add ATP. What if we add CTP? It goes, guess what? That way. Like that. Notice the Vmax and Km are not changing because you're not changing the number of subunits, right? So this is CTP, is doing that. So CTP would be called an allosteric inhibitor, and ATP would be called an allosteric activator. Yes? That's a nucleotide that's at it's way past that reaction that this catalyzes. But if you build up too much CTP, you're overproducing it. So it feeds back and blocks its own production. And it makes a lot of sense. And if you've got a wealth of ATPs, which your cells have lots of ATP, it says it's okay to actually make a little bit more of that carbonyl compound, so it actually stimulates that curve and shifts it to the left. So understand what shifting left and right means and how that influences it. Because we're going to work a problem here in a moment. But one last, so are we clear on what's going on here? Now, let's say, what if we were to break this apart, and we can do this in the laboratory, and there'll actually be a problem when I can here in a moment that will work. And we break it into individual subunits. So we have one catalytic subunit, and we get rid of all the regulatory subunits, and we put that one regulatory subunit in a test tube. What do you think the kinetics are going to look like? Somebody said it. Michaela Smith did. It has nothing that's allosterically informing it or regulating it. So what it's going to look like is this. That's one subunit. If you put ATP or CTP, what's going to happen to that curve? Okay, so would it shift? In which direction? Let's say ATP. Which way is it going to shift? To the left? How is it going to know the ATP is there? There's no ATP, there's no regulatory subunit in the tube. So that's actually, you're correct, you're thinking that, yeah, that's right, ATP is an activator, but there's no regulatory subunit for it to talk to. So it would do nothing. Okay. All right. Oh, and why is the Vmax so much lower than the Bmax? Yeah.
There's only one catalytic subunit.
Right. There's less monkeys in the room. Normally there's 6 monkeys together, and now you cut that down to 1. That makes sense, doesn't it? Okay, good. We learned a lot from that. I hope. Let's take a break.
Yeah.
A little bit more to get through this. Okay. Let's actually go back before we get into the next type of regulation. Let's do some exercises, and actually let's talk just briefly about that. The effect of multiple subunits, regulatory subunits, can also cause sigmoidicity in the inhibition curve. So this is an entirely different plot. It's relative enzyme activity, so it's not an initial rate. And I'm not sure why your book shows this, except that what it's showing is the effect of those multiple 6 regulatory subunits is to increase the effective inhibition power of that inhibitor over very tiny inhibitor substrate range. So in other words, you don't have to have much CTP to turn off that ATC-ACE enzyme. Okay, so if you have just one subunit, it's kind of sluggish, and so the decrease takes quite a bit of inhibitor to cause that inhibition, right? But if you increase, look what's happening. It's very steeply inhibited. This is inhibition, okay? And because this is relative total enzyme activity, so a lower number means more inhibition. And so if you put 4 subunits, it's going to shift it again, it's going to shift it to the left and cause this thing to drop rapidly, okay? But we're not going to talk much about this curve. This is just to show you that the regulatory subunits themselves are cooperating, okay? So there's cooperativity among themselves, and it's positive cooperativity. Okay, let's do some problems, okay? And we're really not far enough along. We're going to pick up the name and the effect of this enzyme here in a couple of lectures, but it doesn't really matter. But the first way to approach an enzyme problem is to know the biology, okay? So you do need to know what a histone deacetylase is. Otherwise, you couldn't understand what the effect of different compounds are going to be on the biology. So we're talking about a tumor that produces lots of HDAC. HDACs aid the tumor in its ability to fold the DNA into mitotic chromosomes. Why does a tumor want to do that? Why would an HDAC be something that the tumor would like to boost the concentration of? Yeah.
Because it wants to just do an abnormal growth of cells, so...
It wants to do nothing
reduce-
except go through mitosis, grow just a little bit, go through mitosis over and over and over. Its favorite job is one thing, divide. That's why tumor cells inside of a normal tissue histologically look like little dots. They're always small. They don't give themselves time to go through G1 and G2 and spend any time there. They don't want to get big. They just want to go mitosis, synthesis, mitosis, synthesis, mitosis, synthesis, okay? And the thing that helps them do that is expression of this enzyme, histone deacetylase. And we'll talk about why that's what a tumor wants. But right now, it's only important for you to understand, number one, this is an allosteric enzyme, and I give you some information about how many regulatory subunits, and I tell you it's an allosteric enzyme. But what you need to know is the tumor wants that. You want to shut that down. Your job is to devise a compound that will shut it down. So that's actually Dr. Connie Valu's job. So now read that question. So this is the paragraph that tells you what's going on here. And it helps to kind of doodle when you're trying to figure this stuff out. Okay? So we got X and Y. This is the question. Now come up with an answer yourself. Okay. And then what I want you to do is to turn to your neighbor and come up with a joint answer. Discuss it. I'm trying to come to an agreement on what the answer I'm sure there is. Why don't you go back and look at that I bet you can't adjust it. But maybe you should just... Is this, oh, this is the old one. There's another background that doesn't have that. All right. Who wants to try that? Drug X, okay.
You want to inhibit the tumor.
Right.
So you wouldn't want HDAC to go in the forward direction or speed up.
Right. You don't want it to be activated.
You would do ATP. Oh, you don't want it to be activated, then it would be...
You want a CTP.
Okay, so then it would be drug block.
It's drug block. So you self-corrected. That's good. Okay, so understand the biology. It's just a straightforward what we talked about. And just don't look at all the babble. There's only a few pieces of information that are important there. Okay. Now let's do another one. Everybody understand that, by the way? Drug one. Because you want to block HDAC. And that actually is a new targeted chemotherapy that I've been involved with, and it works really well. And it doesn't kill a lot of the normal cells. Okay. Oops. And remember, what drug X is doing is it's shifting it to the left. And I'll let you can just, well, take a second and figure it out on your own, and then talk to your neighbor. Or talk to him immediately. Pester them. Heckle. And again, it's really useful, even if you think, oh, this is straightforward, it's possible for you to screw up anyway. Draw it out. Convince the person next to her.
I'm just trying to remember all the stuff looking at it and I realize I see what you promote dealing with application and so tightens the
Okay, I guess people are fairly quick at this, all right? And it was, I gave it to you early last night. Drug X appears to be what type of C, allosteric activator? Any disagreement? No disagreement? Okay, so it is an allosteric activator, even though you don't want it to be, that's what it is, okay? So don't confuse what you want with what's actually happening. And the answer is not E, by the way, but so just ignore that one. Even if you know what a histone code writer is. Okay. Actually, let's do this. Let's go from the bottom up. All right. And so we know it's not this one. She could treat the complex with a low concentration of DNA. Pardon me?
I said DNA is irrelevant.
But actually, it is to the histone. So, but you're right. It's irrelevant to this reaction or to this protein, right? That's what you're saying. But sometimes students mix up the it's a HDAC, and you'll understand very soon that an HDAC will bind to DNA. It's there on DNA, but that's not the function. So that's right. I zero in on the function. She can treat the complex with a high concentration of a kinase. What's a kinase? Phosphorylase. Okay. Is that going to do it? It might change the activity of that one subunit because, you'll see in a moment, that's exactly how the cell does a lot of activity changing in the cell is the phosphorylate thing. But is it going to pull apart all 12 subunits? Probably not. Okay. Treat it with a protease. Well, you want to chop it up. Now that's going to take it apart. But are you going to be able to study it after you've chopped it to pieces? No. So that's not a good idea. So the best answer is urea. And why would you want to use your urea?
Breaks down hydrogen bonds.
Yeah, so it gets in there and it interferes with the non-covalent hydrogen bonds that are holding it together and pulls it apart. Now, if you're going to study it, so you pull it apart with urea, do you have to get rid of the urea before you study the catalytic activity?
Yeah.
Because you've unfolded it, okay? We had a question on an exam with exactly that. What would what would be, if you left the urea in there, you have one subunit in every tube and you left the urea in there, what is the activity, what's the shape of the curve of enzyme activity versus substrate? Flat line. And 90% of the class missed that question. Because they forgot you've got to get rid of the urea because it's just going to keep it unfolded. So don't make that mistake. So these little details, you have to think about what's going on, what's in the tube, how many monkeys are in the room, okay? All right, no good. Okay, here we go. One more. Okay. All right. One thing about this type of question is, and I, like some of my colleagues, I don't try to trick you by burying this word somewhere. I make it big, large, and friendly. Okay. So you need to understand what effect you're looking for the incorrect thing. So there's going to be 4 things that are correct, and you're looking for the one of these things is not like the other. Okay. That's what you're looking for. Okay. All right. Which is the one that doesn't fit? D. D? D. B. Right. Because this is a single subunit. It has no regulatory subunit. A lot of people put something about allosteric activator or something, or whatever, but this is a single subunit that has Michaelis-Menten kinetics, and so it is valid to do the kinetics and ask the effect of what it has on Km and Vmax, but it won't bind to an allosteric site because it's gone. Okay, good. All right. Okay, so we're now exploring different ways that we can regulate enzymes other than feedback inhibitors and things. And a really big one is actually phosphorylating things. That's a molecular switch, and it can be an on switch or it can be an off switch. Okay, so if you phosphorylate things, sometimes they turn on, and then the phosphatase, when it cuts it off, it turns it back off. But it's also possible for the phosphorylation to turn things off. Some proteins are turned on by phosphorylation, some are turned off. So don't assume that if it's phosphorylated, it's activated. Okay, that's a misconception. It depends on the protein. Okay, so let's look at an example where that happens. It's not important to know the reaction too well, but what this enzyme does, this is glycogen phosphorylase B and A. Okay, the B state is inactive, the A state is active. So what it's doing is it's taking, this is a chain, a glycogen chain, it's going to phosphorylate a glucose molecule and put it into one position, and that cuts it off, right? That's what its job is. But the way this becomes active, and you can think of that A state, glycogen phosphorylase A, is active, okay? It's active when it's phosphorylated, and so what happens is there's a kinase that's called glycogen phosphorylase kinase, GPK, that regulates the phosphorylation state of this enzyme. Okay? So it's going to put a phosphate group and convert it from the B enzyme state to the A. So this is this is a poster child of a protein that's activated by phosphorylation. Glycogen synthase, which is an enzyme that takes glucose and sticks them together, is inactivated by phosphorylation. Okay? So that's a good example for that. Okay. Now, how else can we regulate enzymes? And one way is by proteolytic activation or inhibition. Okay, but this is a good example of proteolytic activation. So the pancreas makes lots of enzymes, digestive enzymes, but it doesn't care about digesting itself. Okay, that's called pancreatitis. When the pancreas releases active enzymes into itself, that's a disease. Okay, so what it does is it makes all of these enzymes in an inactive zymogen, it's called. Zymogen state means that the enzyme is inactivated by usually a little peptide overhang that kind of puts its foot in its own catalytic mouth. Okay? And so it's actually part of the protein. And when this is secreted into the duodenum has all kinds of membrane proteases of all kinds. Okay? And what it does is it chops off that little protein overlay and cuts and proteolytically cleaves that enzyme into an active state. And then carboxypeptase starts cutting the proteins that you've eaten and been digested into your small intestine, right? But it also acts on other enzymes and cuts off their inhibition little overhang. And so now this is active, and then that activates this, and so it's sort of a chain reaction of these digestive enzymes. Once you get the first one activated by cutting off that little overhang, the rest of them work on each other. But their job is to do what? Is to break proteins and peptides into individual, they could be individual amino acids or dipeptides. Usually, and there's very few tripeptides. What's the point of breaking it into those little pieces? Why are you doing that? Why not just take up the whole protein? Pardon me? It is. It's much faster to digest single monomers. So what we do, our digestive system goes, it tries to break all polymers, including DNA, lipids, and amino acids and proteins, tries to break them into the monomeric form. Because then you can take a transport protein that we'll study and use it to quickly transport those into the bloodstream. So yeah, that's why you do it. Okay. Good. And then another thing you can do, this is a different type, but it's still proteolytic activation. Now, insulin is not an enzyme, but in general, proteins can be either inhibited or activated by proteolytic cleavage. And insulin is a good example of a hormone that's activated in the secretory granule. We talked about this earlier. So the reason it's made in a proinsulin shape like this is, why do you have all that wasted protein? Well, it's because it folds better in that shape, and then you form the disulfide bridges in the right position. If you put them in the wrong position, you have to uncleave them and then reassociate them. And this is, of course, in the oxidized state, because we're in the secretory pathway. So that means we're either in the ER, Golgi, or secretory vesicle, and that compartment is identical to outside the cell. That's called the exoplasmic compartment. It is an oxidizing environment. So these things form naturally. But when it's sitting there in the secretory vesicle, it does not secrete this molecule. What happens is what's called a prohormone convertase cuts it in two places. And this little shelf lifts off. And it looks like a C, so it's called, guess what? C-peptide. And what we're finding out is, so both the C-peptide and the insulin get co-secreted into your bloodstream. And we used to think that C-peptide was just a wasted protein, right? But what we're finding out now, so what do you give diabetics? You don't give them C-peptide, right? You give them insulin or an analog of insulin, right? But we're finding out that nature intended us to get the C-peptide as well, because it turns out that the C-peptide prevents the growth of blood vessels and their thickening from happening. So insulin's a really good growth factor, but if you get overgrowth of certain blood vessels, they clog up and you get atherosclerosis, okay? So it looks like the C-peptide prevents the inflammation of this action of insulin. So it may be very soon, they're doing clinical studies now, that pretty soon all diabetics that take insulin will take C-peptide too. Okay. All right, so it's not trivial stuff. And the last thing is to just talk about a really obvious, but sometimes it's easy to forget this. One of the most fundamental ways of activating an enzyme or affecting its activity is to control its synthesis. Okay, so you have enzyme concentration. Okay, and so we have two things that regulate that: synthesis, right? That increases enzyme concentration, and we have degradation. Okay, and that, let's give that a red thing. So that decreases enzyme concentration, correct? Okay. Now, so what we have is gene induction. That is, if we stimulate the transcription, what's that going to do to synthesis? It increases synthesis of messenger RNA. That goes out the nuclear pore. It binds to an unsuspecting ribosome. The ribosome turns the messenger RNA into protein, and the enzyme concentration goes up. Right? That's enzyme induction. And so, can we have the opposite effect? So, if you have gene repression, that's going to. So this is a stimulate synthesis, and this inhibits. I'm running out. Here we go. That inhibits synthesis, correct? Now, what's the effect of gene induction on the kinetics of an enzyme if you're measuring that? So let's say the one effect is that you detect that there's increased messenger RNA for that enzyme. What do you predict will happen to the kinetics? Let's say it's first of all, I have to tell you, well, it's a Michaelis-Menten enzyme. What's going to happen?
Speed up.
Be ready. What?
That's speed it up and make more of the enzyme.
It's going to increase the number of monkeys in the room, so what's going to go up? Vmax. That's what I want to get out of it. If you're changing the amount of protein, you don't change the Km because you're making the same protein over and over and over. You're increasing the number of monkeys in the room, right? Okay. Now, so that's going to decrease Vmax, correct? Then there's another side of the equation. It's over here on the negative side. So if you increase proteolysis, okay, and how could you do what's going on, and how can you split up proteins when you, let's say, look, about 10 or 15% of the proteins that are made don't fold. How do you get rid of them? You secrete them? Yeah.
Send them to the lysosome.
Yeah, you could send them to the lysosome. Okay. What if you didn't want to cross a membrane? What is in the cytoplasm that chews up proteins? It's a machine. Lysosome? No, that's a membrane-bound organelle. This is in the cytosol. It's called the proteasome. Okay. The proteasome. It's a shredder, right? Anything that has ubiquitin attached to it, which is a protein, that's a tag that sends it to the shredder. And then when that happens, what's that going to do to the Vmax? go down, less monkeys in the room, right? So it boils down to the monkeys in the room, okay? And then, of course, insulin, one of the ways it works is to inhibit proteolysis. So that's actually going to inhibit, so this is actually, I should have put a green arrow here, okay? Because that's going to stimulate degradation. But if you inhibit proteolysis, that's going to do what? Inhibit degradation and increase the number of monkeys in the room. So it doesn't have to be synthesis, you can just stop proteolysis. And the amount of enzyme will increase because there won't be the degradation that normally occurs. So even though this is it's straightforward, it can be confusing. So don't be. Look at this carefully. In none of these situations would you see a difference in Km. And just the last 30 seconds is how do you expect to get a difference in Km in a biological situation? What would a cell really need to do to make an enzyme with a different Km? Remember what I said about hexokinase? It comes in more than one flavor. Yeah, a different isoform. It would need to be a different gene, or it could be spliced differently. Okay. So that would affect Km. All right, good. Thanks.